I am Dr. Syed Kashif Haleem, Associate Professor Microbiology, Hazara University, and this is a short documentary on my research project, age-related variations in the activation of complement system against Pseudomonas aeruginosa, which was funded by Higher Education Commission of Pakistan. It all started in 2018 when we published our work in Central European Journal of Immunology, and that work demonstrated age-related variations in the bactericidal activity of serum against Pseudomonas aeruginosa. The children and elderly sera were compromised in killing Pseudomonas aeruginosa. So that was interesting, and our hypothesis was that these differences are because of complement system. As we know, in serum we have extracted all the blood cells, so only thing left behind is complement system and complement protein along with the plasma protein. So regarding immune system, the complement protein have the ability to form membrane attack complex on the bacterial surface and kill the bacterial cell. So we thought that these differences might be at level of complement system. In children, they might be due to maturational deficiency of some complement protein, and in case of adults it might be because of age-related dysregulation of some complement protein. So we wanted to investigate this further and identify these proteins which were responsible for a lower level of killing of serum collected from children and elderly. So after execution of the project, we got some very interesting results. By the end of first year and start of second year, we were able to identify the molecule which was responsible for reduced killing of elderly sera, and that molecule was colectin-11. Colectin-11 is the molecule that activates the complement by lectin pathway. We discovered that the binding of this molecule was weak when we used the elderly sera. And because of that weak binding, we further checked that complement activation and C3 deposition was also weak in the elderly sera. So this was the molecule which was responsible for reduced level of killing by elderly sera. And besides that, we also discovered another finding which was not relevant to this project, but that was interesting that we got during this project, and that was CL11 binding and complement activation as a whole was higher on multi-drug resistant strain of Pseudomonas aeruginosa. So that was very promising that we can use complement system as immunotherapeutic potential. And we discovered that higher complement activation on this pathogen is because of the same molecule, the colectin-11, because colectin-11 binding is high on Pseudomonas aeruginosa as compared to the normal wild type strain. And until the second year, it was confusing that CL11 binding was weak in elderly, but it was okay, it was normal as compared to adult in the children's sera. So until the end of study, we were confused that what was the reason of reduced serum killing by children sera. And by the end of third year, we discovered that reduced level of killing of Pseudomonas aeruginosa by children's sera is because of another complement molecule, C9. C9 is the molecule that helps in the formation of membrane attack complex, which is the hole on the surface of Pseudomonas aeruginosa, and because of that hole, the bacterial cell is lysed. And we discovered that CC9 deposition was very weak as compared to other two groups in children's sera, and that might be the reason that complement system cannot induce the formation of membrane attack complex, in simple words, the hole on the bacterial surface. So that led to reduced level of serum mediated killing of Pseudomonas aeruginosa. So in a nutshell, we recommended that C9 and CL11 are the two relevant complement recognition molecules that are responsible for reduced level of killing by children and elderly sera. So after successful identification of these two proteins, we want to carry this study further in future, and we will check that if these two proteins can enhance the immunities of the susceptible groups, children and adults, we can express the recombinant version of these proteins outside by genetic engineering, and we can check that if we inject C9 in children, So can the immunity be enhanced in children? And on the other hand, we can use the recombinant version of colectin-11 as a double-edged sword. Number one, to increase or enhance the immunity of elderly. And number two, the new finding that we got during this study is, can we use colectin-11 to enhance the immunity against multi-drug resistant strain, which is the major concern after the antibiotic resistance these days. So these aspects can be studied, and we are planning to apply for more grants in future to carry out some in vivo studies and to check the efficacy of these molecules in future.
Hello, I am Dr. Isfahan Tosif. I am the chairman of Department of Microbiology, and I was part of this project as co-principal investigator. I would like to highlight 3 key points of this project. Firstly, this project provided valuable opportunity to our research students to engage in high quality research. Through the supreme guidance of Dr. Syed Kashif Haleem and his thanks to his expertise in the field of immunology, our students have been able to conduct research that contributes to the advancement in the field of immunotherapeutics. Secondly, thanks to the wide scope and resources made available through this project, we were able to supervise 1 PhD and 7 MPhil students who completed their research under this project. Lastly, this project also indirectly benefited other research students of the department. 2 additional PhD students also got benefit from this project. This project empowered and inspired the next generation of research students who were enrolled in our department and at this university. It's been a journey of growth, collaboration and innovation, and we are immensely proud of the achievements and contributions made by our students and faculty. Thank you very much.
I am Umair Ahmed. I am PhD student leading this research group. My research analyzed how complement activation varies among different age groups. I also investigated how these variations affect key functions like immune cell engulfment, also known as opsonophagocytosis, and direct killing of cell by formation of membrane attack complex.
Hello, I am Sharman and I am thrilled to take you on a journey through an extraordinary project under the visionary guidance of Dr. Syed Kashif Haleem. My research focus was to find the in vitro binding of a complement lectin pathway activation molecule MBL to Pseudomonas aeruginosa. During my research, I investigated the role of MBL against wild type, different clinical strains and MDR strains of Pseudomonas aeruginosa. And we have found that there is no role of MBL against Pseudomonas aeruginosa. As an operations leader in a multinational company, the lessons from this project resonate profoundly. It's not about what I learned during that project. It's about how those lessons continue to shape my approaches to the challenges. I extend my deepest gratitude to HEC for their support and of course Dr. Syed Kashif Haleem for his unwavering mentorship. Thank you so much.
I am Dharav. My investigation compared complement activation in wild type and MDR strains of Pseudomonas aeruginosa bacteria by analyzing C3 activation patterns.
My name is Ziaur Rahman. I explored how 5-colon L, H and M binds to Pseudomonas aeruginosa.
I am Kashif Haleem. I studied the role of C1q in activating the classical pathway of the complement system against Pseudomonas aeruginosa.
I am from Anali. My research investigated how colectin-11 trigger lectin pathway on the surface of Pseudomonas aeruginosa.
I am Faimullah. My research focused on the formation of membrane attack complex on the surface of Pseudomonas aeruginosa. I investigated how membrane attack complex patterns relate to the killing activity of serum.
I am Matiullah Al-Kozair, an international student from Afghanistan. My research compared the relative effectiveness of 3 complement activation pathways against Pseudomonas aeruginosa.